So for extraction of DNA, we are using this DNA mini kit from Kaizen. It comes with these components. So first is the mini spin column. Second one is the proteinase K. This proteinase K is for denaturing the proteins which are there with DNA. Third one is the buffer AL. The buffer AL comes with guanidine chloride. Its function is basically for lysis of the cell and denaturation of the organelles. And then buffer AW1. Buffer AW1 is reconstituted with ethanol. AW2, these both are wash buffers, and the function of these buffer is to filter out the impurities. Next comes the buffer AE. It is the elution buffer. It will elute out the DNA which is present in this spin column. It is the last step. So few more things are required for this experiment which we have prepared. We need tris buffer, we need EDTA buffer, and from that we prepared the tris acetate EDTA buffer, and we also have prepared the 6-axis loading die which is used while loading the DNA. So before the experiment you must organize your decks, your working decks. Okay, so first you wear your personal protective equipments because you'll be dealing with carcinogenic substances. First take one micropipette, 1.5 ml Eppendorf tube. Then you will add 20 microliter of proteinase K. 20 microliter proteinase K in the pipette. To this we will be adding 200 microliter of the sample. This is the sample which was collected in our EDTA tube. So we are adding 200 microliter of the sample to the proteinase K. Then we'll be adding 200 microliter of AL buffer, which is the lysis buffer, to the sample. Okay. Then we are mixing the sample in a vortex mixer for 15 seconds. So we'll be incubating the sample at 56 degrees centigrade for 10 minutes. This is to ensure that the lysis is proper. So for 10 minutes we'll be incubating the sample at 56 degrees centigrade. Then we will briefly centrifuge it so that the particles which are stuck to the lid will come down. We'll briefly centrifuge it. Okay. Now we'll be adding 200 microliters of ethanol. Now we will be adding 200 microliters of ethanol to the sample. Ethanol will dissolve the DNA. Now we will be mixing it in a vortex mixture for 15 seconds. After mixing, we will then briefly centrifuge it at 800 rpm. Now this is a spin column. Okay, we will be carefully applying that mixture to this spin column. Care should be taken that we do not disturb the rim. We are transferring the sample to a spin column. Again we will centrifuge this for 3 minutes. transfer the spin column carefully into a 1.5 ml microcentrifuge tube, and then we will discard the filtrate. To this, we will add 500 microliter. To this, we will add 500 microliters of buffer AW1, which we have prepared. This is the buffer AW1 we have prepared with ethanol and collected in another container. Buffer AW1, 500 microliter.
No.
Again, we will centrifuge and repeat the process. This is basically we are washing the lysed sample. So we have centrifuged this. Again, we will discard the filtrate and into a clean micropipette tube. We are placing it again, and we will add 500 ml of wash buffer 2, that is AW2, which also we have prepared. We'll be adding 500 ml of this wash buffer 2. Again, we will centrifuge for 5 minutes and another 5 minutes. After centrifugation, we will put this spin column into a clean tube and will discard the filtrate. Then we will be adding 200 ml of AE buffer, 200 ml of AE buffer is added, and then we will incubate the sample at room temperature for 1 minute. Timer is set for 1 minute. After incubation, we will again centrifuge it for 1 minute. After centrifugation, take out the tube carefully. Now this filtrate is our sample. Then we will throw away the spin column. And then this is our sample, DNA sample, which we are going to load.